Ma'am, you can start, ma'am.
Okay. A very good afternoon to all the participants and the dignitaries. On behalf of the Department of Biotechnology, Guru Nanak College, our dynamic management to this afternoon session of Fermentation webinar series. The speaker of this session is Dr. Samuel Jacob, and I welcome you, sir. Now, I request our Vice Principal, ma'am, of Shift 1 to deliver the felicitation address.
Thank you, Dr. Bharati. Good afternoon to our respected resource person, Dr. B. Samuel Jacob from School of Bioengineering, SRM Institute of Science and Technology. Honorable General Secretary and Correspondent of our Guru Nanak College, Mr. Manjit Singh Nair. Most respected Principal, Dr. M.G. Raghunathan. Vice Principal, Shift 2, Dr. Gunita Arun Chando. Dean of Sciences, Dr. Noor Jahan. Head of the Department of Biotechnology, Dr. Bharati Ravi Krishnan. Dr. Mahendra Kumar, Dr. Mahalakshmi, Mr. A. Amala Luduraj, Deans of all schools, heads of all departments, and our special invitees, the participants from all colleges across our nation. All of us have been in the same boat. Yes, we are quarantined, we are in lockdown, but we need not literally lock down our minds. In the available time, let's enrich ourselves, redefine ourselves, and upskill ourselves. Our Guru Nanak College has unlocked the hidden potentials of all departments. As of now, we have crossed 140 programs through webinars, workshops, quiz, FDPs, and the like. Today, the Department of Biotechnology has initiated a webinar series on fermentation technology. I take this opportunity to thank our General Secretary and Correspondent for his constant support, our respected Principal for his consistent motivation, our Vice Principal Shift 2 for her encouragement, Dean of Sciences Dr. Mujahab, Deans of all schools, heads of all departments, members of staff and students, and above all, the learned members of staff of participating colleges and all students who are attending this wonderful program. Let's all make use of this golden opportunity to enhance our knowledge on fermentation industry and its innovation. On behalf of our Guru Nanak College, I heartily welcome our respected resource person, Dr. B. Samuel Jacob sir from SRM Institute of Science and Technology. Hearty welcome to you, sir, and hearty welcome to all the participants. Thank you very much. Over to Dr. Mahendra Kumar.
Thank you, ma'am. Thank you, ma'am. It is my immense pleasure to introduce the speaker to you all. Dr. Samuel Jacob has completed his BTech in Agriculture Biotechnology from Center for Plant Molecular Biology and Biotechnology, Agriculture College and Research Institute, Tamil Nadu Agriculture University, Coimbatore. He has completed his MTech in Industrial Biotechnology at Shastra University, Tanjore, Tamil Nadu. And he has completed his PhD in Microbial Biotechnology in the field of Agriculture and Food Engineering Department, Indian Institute of Technology, Kharagpur, West Bengal, India. He has worked as Senior Fellow in a project at Agriculture Food Engineering Department and PK Sinha Center for Bioenergy, IIT Kharagpur, West Bengal, India, and this is sponsored by Oil and Natural Gas Corporation Energy Center, New Delhi. At present, he is working as assistant professor in the Department of Biotechnology, School of Bioengineering, SRM Institute of Science and Technology, Kattankolattur, Kanchipuram district. Dr. Jacob has 15 research papers and chapters in Scopus Industries, 4 patents, 18 international conference publications to his credits. He has been credited with 3 Best Awards at international and national conferences across India. He mentored 2 innovative product developments, namely IBIN Intelligent Dustbin Technology and Vayu, that were officially selected as national level top 20 teams of biotech entrepreneur students team competition, Best 2018 and 2017 respectively, conducted by Association of Biotechnology led enterprises jointly by DBT Government of India. The innovative biogas Kisan Vayu held at fourth position, top five in the Project Show exhibition competition conducted by the Institution of Engineering and Technology on campus, SRM Institute of Science and Technology, Chennai. Further exhibited the model at an industry institute summit conducted by the Institution of Engineering and Technology, Chennai. He is currently working on various waste valorization technologies and futuristic concepts of solid waste management. Teaching expertise include bioprocess engineering, microbiology, environmental biotechnology, industrial microbiology, and industrial fermentation technology. Today, he is going to share his expertise on upstream process in fermentation industry and its innovation. Now, I welcome Dr. Samuel Jacob sir to share his expertise among us.
Thank you, ma'am. Thanks for the nice introduction. Thank you, Vice Principal, for giving me the introductory session. And yeah, welcome you all. And I'm happy to receive you today for my webinar session. And I'm going to discuss about, and I'm going to share my thoughts and expertise upon the upstream process and innovation that was required for a fermentation industry. We'll first go into the yes. Without fermentation, almost half of our products that are considered to be important in our life will be not there if there is no fermentation. Say, for example, it starts from food that we daily take. Start from the curd to pickles and everything that we take out of the fermentation only. And the other extreme, if you see the health products, mostly the antibiotics that are now the life-saving phenomena in our whole world is from the fermentation process. And you know, as these milk products, deliveries are all part of the fermentation. So we can assume that without fermentation, nothing is being much possible to meet our daily to day needs. Yes. So that's why there is a huge demand in this field and a continuous upgradation and innovation being required for fermentation industry. So today I'm going to see how a fermentation process looks like and how what is the upstream processing in the fermentation industry, how it is more important towards the commercial viability of an industry, what are the innovations required by the industry. Okay, before when going into it, I'll just give a little glimpse and interesting facts about industrial fermentation process and its origin, so that you can realize where from where from we started and now where we are with the developed technology. Yes, the origin of industrial fermentation started in the 17th century. The scientific industrial fermentation. Okay, this started by George N. Stark. Okay. So he is the first person who coined the term that fermentation, where the some chemical synthesis is going on, recombination is going on to convert a product substrate to a product. Say, for example, sugar converted to an ethanol. It's a chemical recombination or chemical fermentation technology as a coined word. But it's not about 17th century. Even before, it's about 2160 before Christ. The Egyptians and Greece people were well aware of this fermentation technology, but not about the scientific background behind it. Say, for example, you can still find from the picture, you can still find in Egyptian pyramids and all, there are some sculptures that depicts the wine prominence among the kings and queens. Okay, but that time, what they were thinking is that the transformation of wine, a grape juice into a wine, is something by a uh, godly creation or something a godly activity. Because as you know, these Greek people have a lot of gods: God for water, God for fire, God for land, okay, God for air. Also they have. So similarly, they thought that this transformation is being done by some godly activity, and hence it's a beverage consumed among the elite people in the Greece and the Italian Roman region. Okay, yes, it's not about the 17th century. It's been done even before, and they have been prominent in doing the bread making also. Okay, these are some of the historical phenomena that we can realize where from this fermentation technology has started. And yes, it has been started to are geared up to have a very much high potential towards the human beneficiary once then Louis Pasteur started to delineate or identify this chemical transformation or recombination first that has been done by Ernest, that it's done by the microorganism. So he is the first person to identify the microbiological background behind this fermentation. So Louis Pasteur, as many of you know, right? Louis Pasteur is a French scientist. So he is the first person, and he is the first person to coin the technology called pasteurization. So he declassified the microorganism into two different ways. One is the beneficial, and another is a spoilage activity. So we have two different groups of microorganisms. Say, for example, you take a milk and add a small quantity of curd, keep it in the kitchen. Next day, what happens? The milk converts into curd. Curd again, we are taking it. The same milk without adding curd, just keep it as such in the kitchen. Next day you come, it also formed as a curd, but you cannot able to consume because it is being populated by different spoilage microorganisms. So that's the difference between the microorganism that is acting a different role in the same substrate itself. So beneficial, say for example, same example, if I can tell, the milk, the lactobacillus, the beneficial lactobacillus only converting the milk, the lactose into the fermented curd. Okay, whereas the other species, Staphylococcus and everything, Streptococcus, are spoiling the milk. Okay, so he declassified. So how to get rid of it? Even the raw milk, once extracted from the cattle, has to be pasteurized because it is not very highly sterile once it's from the cattle. So it has to be removed of other pathogenic microorganism or spoilage causing microorganism to convert into a consumable product. This first has been not in the milk. He was first to implement this in a wine because Europeans, wine is one of the part and parcel of their daily diet. Like how we are taking milk in our daily diet, for them, wine. So the wine consumption is more uh, important for them. So the wine, for fermentation, he postulated the pasteurization technology to avoid the spoilage causing microorganisms. Okay. Yes, you can see that how the history has been evolved of initial fermentation. You can see a lot of wooden barrels. Yes, that's how the reactors or those initial days are not made of the steels or stainless steel that are present today. It is all made up of wooden only. Even now, traditional European winemaking companies, in the like how we have a cottage industry, they have a cottage industry that still make the wine in the wooden drums. Okay, so this is how the development has started. Early days of industrial fermentation has started. They slowly from winemaking to they stepped on to a beer making, the diversification of products. The from the same substrate, wine, beer, whiskey, brand, like different breweries. Why I'm telling these example of breweries is that this brewing is making a huge market among the fermentation industries in globally. Yes, fermentation still get geared up when the yeast yeast cell has been identified. High efficient yeast has been identified, and Emil Christian Hansen, he is the first German scientist, and he is in Denmark, and he identified this Carlsberg yeast number one, patented it, and it's still now the world prominent beer producer in the world, Carlsberg beer. Okay, so why I'm telling is that single strain that he identified, the pure culture is that he identified and made it as a more more robust, so that it can withstand high ethanol concentration because ethanol concentration can kill a microorganism. See now what we are doing, we are using sanitizers for sanitizing our hand. Sanitizing means for removal of microbes from our hand. So what the sanitizer contains? It contains ethanol. So ethanol is actually can able to kill the microbes also. So you should find the appropriate candidate to withstand such ethanol concentration. This Hansen has identified this Carlsberg yeast number one, which is patented one, and it's been prominent among the fermentation industry, especially in the beer market. Slowly, in the late 20th century, that need-based development has started to emerge. Okay, so it's the diversification of fermentation different ways. Say, for example, in environment, the treating of water, the wastewater to convert into a purified water or semi-treated water, so that if we release into the environment, it won't hamper the environment. That is the other environmental pollution. So here also fermentation plays a huge role in the environmental aspect. And another significant area is that healthcare segment where the antibiotics that I mentioned before playing a prominent role in the fermentation industries. We'll be seeing in one tool. So when I tell antibiotics, first thing anybody or any graduates or any person, even school children, name in one antibiotic, first thing they will tell about penicillin. Yeah. So the penicillin is the widely used antibiotic in the early 20th century, especially during the World War time. Okay, so you know that Alexander Fleming, 1940, has identified and accidentally discovered. It's not very targeted discovery of penicillin. He just doing an experiment towards Staphylococcus infection, and he found a contamination in his plate. You can see the plate here itself. So the white, the streaked one are the Staphylococcus states, and you can see the cottony patch that is nothing but the Penicillium rotator. Okay, so the Penicillium, when it is growing, it has hampered the growth of the staphylococcus, which means that it is a bacteria and it is a fungi. The mold is a fungi. So something is produced by the fungi that hampers the growth of the bacteria. So it become an antibiotic. Okay. So slowly he started to reverse the research towards the cottony patch that is on the penicillium, and he ended up in identifying the penicillin that played a huge part in the World War because the war zones required a lot of antibiotics for the curing of the wounds caused in the warfare. Okay, so it has a huge boom like for in a fermentation industry, and slowly the citric acid production and other organic solvent production has taken its slow initiation in the early 20th century. Okay. And now recent advancement has emerged that has pushed the limit of fermentation application in the healthcare sector, especially in the monoclonal antibodies for the therapeutic, diagnostic, and other analytical purpose. Say, for example, now the present situation, what we are today is the pandemic caused by the COVID virus. Okay, so now people are how they are doing. They are doing PCR reactions, right? PCR reaction, and they were available in the Chinese sector also. Now Indian failure. So the kits are made up of the kits are made up of the antibodies. Okay, antibodies against the particular the viral infection. So to produce such a huge amount of antibodies, you need a fermentation process to be carried over for mass production. And now recently, genetic engineering aspect has been employed for novel fermentation process. Even the insulin that we take today is not an insulin from the human. The insulin is a recombinant insulin that is called humulin, that is obtained by genetic engineering approach, and we are using in our daily today life. You know, the prominent leader in the humulin production is Biocon, that is from India. But you all know the Dr. Kiran Mazumdar Shah is the first women entrepreneur in India to have a whole pharmaceutical industry that is mainly based upon the genetic engineering of the yeast. Okay, so these are some of the periodical timelines that how the developments of industrial fermentation started. Yes, now we have ended up into this much diversified field and different products, starting from food to the industrial chemicals, fuels, microbial enzymes that has a different applications are all being the products of fermentation. Now the burden on us. Burden on us means burden on the biotechnologists, how we are able to withstand this much demand, like to have this much diversified product or this much diversified application, how we are going to act towards it. So that's why you need a routine, or you need a constant or frequent upgradation in this technology. Especially nowadays, you know the environment. Environment given us more emphasis because we are in the climate change, environmental pollution is happening. How to deal with environmental pollution, all dependent upon the what type of technology we are going to implement for such activities. Okay, we'll be seeing like among this how we are going to implement it. Okay. So fermentation means always we look into it. What the microbe does the fermentation? No, it's not so. Microbe is one of the candidates that we are using in the fermentation process. Even animal cells and plant cells are also being used in the fermentation industries. Say for example, you heard of plant tissue culture, right? So in the plant tissue culture, still we can delineate into the plant cell cultures where we can able to produce the plant metabolites from the callus culture of the plants. And animal cells are also being used for the production of metabolites from the animal cells. But there are some advantages that microbes that afford us so that we can able to employ the microbes in a large scale. And some of the advantages of plants and animals can also being utilized in the microbes through genetic engineering approach. Say, for example, some metabolite is produced by the plant cell in the fermentation process. Instead of, but you find some difficulty in using plant cells. So what we can do is that we can take only the gene of the plant cell and clone into a microbe and employ in the fermentation process. That's how the insulin that is being produced. Same thing, animal cells, that insulin from the animal being gene coding for the insulin is being taken up and flown into a yeast microbe and being produced in the basket. Yes. So let me tell you why the microbial fermentation play a prominent role and why it is more economically feasible in a production of lot of commercial products. So it is only because of the ease of their mass cultivation. Microbes are very easy because okay, whereas the plant animals are very tender and very sensitive towards the cultivation and the speed of growth. The microbes grow multiplies very fast. Like the least that is that E. coli multiplies every 20 minutes. Any biotechnologist can tell without any thinking the doubling time of the E. coli is the 20 minutes. So speed of the growth. Even the microbes are even fast growing less than that too. But we know only the yeast now. So use of cheap substrate, okay, especially the plants and animal cells requires hormones and other costly enzymes and other components in the media that increases the cost of the product, okay. Whereas the microbes can able to survive can able to metabolize this in a cheap substrate, sometimes from the waste material also, so that the cost of the product, when the cost of production is higher, the product cost is also getting higher. Okay, that is the economics behind it. When the production cost is more, the cost of sale is also more. So in order to reduce the cost of sale, they have to reduce the cost of production. So only through the advent of this microbial fermentation, they can able to produce the product with lesser cost and diversified potential products. As I told, even the plant and animal products can also be tapped or can also be cloned in the microorganism to produce. Okay, not limited to the alcohol. Even the organic solvents can also be produced by the microorganism and our ability to undergo genetic manipulation. As you know, where microbes are very easy to handle the genetic modification. You can either use the gene gun or electroporation or any other method that is very easy to are very amicable to undergo genetic modification. That's why the microbes are playing a prominent role in the fermentation industry. Okay, so before now we are slowly entering into our today's topic, right? So far we have seen about the importance of fermentation. Slowly we go into the fermentation, declassification fermentation. As you know, the microorganism, right? So we are going to concentrate about the micro fermentation only. So the microorganism has a sigmoid growth curve. Many of you are aware of that. There are different segmentation in the growth curve. Starts from log, and stationary. Why this growth curve is playing a significant role? This is like a blueprint for a industry or blueprint for how a blueprint plays a major role in a building. So similarly, the blueprint of a microorganism growth curve play a significant role in the product production. So we classify the products into two different. One is the primary metabolite and second is the secondary metabolite. Primary metabolite are produced during the growth of an organism. So when the microorganism grows, some of the metabolites are produced constantly. Say for example, ethanol, okay, other organic solvents, the TCA products like citric acid cycle products are all the growth as well. Whenever the growth happens, these products are released into the bulk media. Okay, whereas the second classified product is the secondary metabolite. Metabolite, the chemical that is produced for the metabolism, that is called metabolite. It's a non-growth associate. Even then there is no growth occurring, these secondary metabolites are also produced. Mostly, the secondary metabolites are produced by the microorganism for the survivability or sustainability. Okay. Say for example, here lag phase is the adaptable phase for the microorganism. Once you put the microorganism in a media, it does not go initially. Okay, it takes some time to adapt to a new condition and then grow. So the time taken by the microorganism for adaptation is called lag phase. Once it get adapted, it started to grow in a logarithmic phase. Logarithmic means doubling. So microorganism grow in the binary fashion. Okay, the doubling of logarithm. So it has a steep increase. So when it is steep increase, it goes very fast. More faster it grows, more synthesis of products takes place. You can see the curve happening, increase in the product. Okay, and then once the growth happens, growth where the growth stop means once the nutrient available for the growth started to extinguish. Okay, say for example, a carbon source is provided as a nutrient media in the nutrient media. When the carbon source is being utilized completely by the microorganism, then the microorganism will be divided because multiplication happens very fast. Population is more, and the sugar start to, I mean, the carbon source start to reduce. Once the carbon source started to distinguish or carbon started to extinguish, so it attains the stationary phase where there is the very less carbon source, more microorganism, and the microorganisms started to fight with each other for the meager amount of carbon source available. So microorganism has to survive under such environment. So what happened is that we are having the brain, we are having the hand, we are having the legs to fight with each other to get a product of our interest. Okay. Whereas the microorganism does not have. What it has to do is that it has to act in a chemotaxis. Chemotaxis means so it has to produce some chemical to accustom to the particular environment. So it has to produce a metabolite. Say for example, the antibiotic, the penicillium, and other streptomycin, and all the antibiotics that are produced by the microorganism to kill the other microorganisms. Okay, to so that it can survive. Okay, it's a kind of evolution only. So different microorganisms produce different secondary metabolites. So this is the blueprint of the even some more secondary metabolites. Say for example, even the pigments. Okay, the pigments are all produced by produced by the secondary metabolite. Okay, so these are all the some of the glimpse of the primary secondary. Okay. So as I told, primary mostly the ethanol, lactic acid, and butanol are the examples of the primary catabolic products. And anabolic products. Anabolism is that catabolism is breakdown of a larger molecule into a smaller molecule. Say for example, glucose is being broken down into ethanol, lactic acid, and butanol. That is a catabolic process. The opposite of it is the anabolic process. So combining the small molecules to form a bigger molecule. Say for example, from the small nitrogenous components, from the small amino acids, slowly the enzymes and other nucleic acids started to emerge. Okay, so this is an anabolic and catabolic products that we have for our daily to day life. These are some of the examples that you can, if you can browse also, you can able to get some of the anabolic and catabolic products, and these are the most commercial products that are only applying to the microbial fermentation. There is no much of chemical alternative routes for production of this type of products. Let us come to the secondary metabolites. As I told, secondary metabolites are not only produced by the microorganism, secondary metabolites are produced by the even the plants also. Say for example, the color of the flower. Okay, the flower color does not play any role for the plant's growth. Okay, that is only to attract the insects to have a pollination. Okay, so even in the color of when the color of the flower is not there, then also pollination happens sometimes. Say for example, you can see the white flowers also. They are not colored, but still pollination happens. How it can able to produce a fragrance? Okay, the fragrance is nothing but a volatilizable secondary metabolite. So these pigments, fragrance, fragrance producing components, flavors are all the components of secondary metabolites only. Now you can analyze the whatever the food industries that we use. Say for example, when we take the 7-Up or Sprite, we get a lemon flavor. That is not really from the lemon. That might not be directly from the lemon juice. That are now also being synthetically produced by the microorganism citric acid and being used in the beverage industries. That's why I'm telling that these secondary metabolites play a significant role in the fermentation industry, especially in the food beverages. Okay, so even the clone that are the clones of the plants can also be genetically recombinant with the microorganism to produce the commercial product. Now let us come to the microorganism secondary metabolite. So whether all microorganisms produce secondary metabolite? No, not all microorganisms. Some microorganisms produce secondary metabolite, especially bacteria. The secondary metabolite is governed by the not by the chromosomal DNA. It is by the extra chromosomal DNA called plasmid. Okay, when there is no plasmid, there won't be any survivability or sustenance for the microorganisms. So this plasmid is helpful for in two ways. It helps us to produce the antibody by the microorganism, and this plasmid are also being used for the genetic recombination that you know already better. Okay, I'm not going into the topic, and it is making us more useful for both production process and also the genetic modification purpose also. Okay, so that's how we are tapping the secondary metabolite production in the microorganism. These are some of the secondary metabolites that are having the mostly the pharmaceutical application. Okay, so because antibiotics, I told penicillin, streptomycin, actinomycin, bleomycin are all the antibiotics that are produced in the fermentation industries and applied for the clinical use. And as I told, you can see in the last portion, you can see the food flavor production from the even from the microbial second metabolites too that are being commercially exploited. Yes. So when you can remember the last two, three slides before I told, the production cost play a significant role in the sales cost of the product. So fermentation industry has classified the whole product into two different types. Whole industrial fermentation industry products are being narrowly being classified into two. One is high volume, low value products. The production quantity is higher in the fermentation industry, and the product cost also very low. Say for example, ethanol. Okay, so because ethanol, they don't want, so it won't be sold as a higher cost. You can see, right? Now day to day life also you can see. Okay, whereas the low volume and high value products, some products are being produced in the low volume. Say for example, hormones, antibiotics are not produced in a large quantity. Okay, so that are produced in a small quantity, but its cost is higher. When you go and get a insulin shots at a more higher cost as compared to ethanol. That's because of the intensification or the production that happens in the fermentation industry. Okay, so generally the secondary metabolites are being produced in a very small quantity. Another is classical example I can tell you that many of you can correlate. Secondary metabolite is that the venom of the snakes. Okay, the snake venom. Okay, snake venom. Even what is snake venom? When the snake realizes that it is under an uncomfortable condition, what happens? It bites the person who disturbs and injects it a small quantity of venom. What happens at venom? Even though it is small quantity, it can cause a huge reaction inside. Okay. So similarly, the antibiotics are also similarly a small quantity have a huge reaction. Especially these are being produced in the small quantities, even in the nature also. Okay, so that's why they're classified into two different ways. So when you want to start an industry, if many nowadays many entrepreneurs are blooming up, even in the fermentation industry also. So you need to target what type of products you want to going to produce. Okay, so again, what type of product means depending upon the market value, depending upon the market demand. From this, you can choose it as a whether it is high volume, low value product, and low volume or high volume. So recently, if I can tell a classical example of this beverage industry, nowadays there is a lot of brewing of non-alcoholic beverages. Okay, so previously people are not willing to take the beer, whiskey, brandy, and all because these are comes under the alcoholic beverages. Now recently there was a small innovation happening behind the fermentation industry. Why not to produce a non-alcoholic beverages? So that's how being now brewing up. Okay, so that I will be discussing in later sections. So what are the innovations that are being expected out of it? Okay. So what are the characteristics of the microbes? Even though microorganisms are being suitable candidate for fermentation production, what characteristic you need to have to employ it as a character? Because all microorganisms cannot be used for the fermentation industry. Only few microorganisms or only some microorganisms we can choose it for commercial exploitation. So on what basis we need to choose that microorganism? On what basis? On what characters of the microorganism we need to select it for the fermentation production? Okay, so we'll see one by one. This is more important, especially in the upstream processing. Before you start an industry or before for a in the industrial aspect, need to know what microorganism you need to choose. So the microorganism must be able to grow in a simple medium. As I told before, simple medium means it can able to grow in the waste also. Okay, so that's why it should not require very robust or very costly chemicals required for its growth. Otherwise, your cost of production will be higher. Okay, so that's what that growth medium and the cost is always interlinked with each other. Okay, so and third thing is that the microorganism has to grow vigorously and rapidly. Because why we are going to microbes? Because it can able to multiply much faster as compared to plant and animal cells. So some microorganisms not always result in a very rapid growth. Say for example, anaerobic microorganism like methanogens and all those, it has a doubling type of more than 48 to 72 hours. Okay, so that is a huge, especially the microalgae. So microalgae is also run the cyanobacteria and microalgae. The W time is much longer. Okay, so you should not choose that because the more the time it takes, again more cost for electricity production or electricity utilization for the fermentation production. Again, this cost also being reflected in the cost of production. Okay, so the microorganism has to grow very fast and vigorously in the medium, and it has to produce the desired materials in a required quantity. So it should not like, even though ethanol is being produced by different microorganisms, it should be produced in a adequate quantity that can be commercially utilizable. Okay, it should all micro some microorganism produces ethanol also, but it is not in a much quantity, so we cannot use it for a commercial purpose. We can use it for a laboratory and other purposes, but we cannot exploit for a commercial purpose. So these are some of the important aspects for considering the microorganism to be used in as a industrial strain. Okay. And another important thing is that microorganisms should not produce toxic or undesirable materials along its production of commercial products. Say, for example, when the microorganism producing ethanol, it should not produce another product that are poisonous or toxic in nature. So we should ensure that the microorganism is not producing any toxic components or undesirable material. Otherwise, we need to have a robust, or we need to have a very uh, high purification technique that will also incur more cost. Okay, to purify the product from the toxic and undesirable materials. And microorganisms should have a reasonable genetic and physiological stability. Yes, the microorganism, as I told, it undergo very rapid cell division. The more the cell division, the more the probability of mutation. Mutation, you know what is mutation, right? So when there is more division happen, there is the crossover happens in the genes. So what happens? The high producer microorganism might lose its property of high producer into a low producer or medium producer. So what happens with increasing time? Maybe initially the strain might be suitable for production of large quantity of product, but later what happens due to mutation and genetic instability and physiological instability? What happens? The product quantity started to reduce, sometimes quality also. So we should realize, or we should consider this very much essential for our fermentation industry. As I told before, Hans Hansberg is number one. Okay, Carlsberg Hansen invented Carlsberg is number one. That has been done in the 19th century. Still, that is the world prominent producer. Why? Because their strain is more genetic and physiological stable for more than 100 years. Okay, more than a century, they can able to uh, they can able to maintain the stability. That's why they can be a world prominent leader in the beer production. Okay, and at the end of the production, it should be very easy to harvest. Easy to harvest means the cost of harvesting, or when it is reduced, obviously the product cost also will be reduced. Okay, and microorganisms should able to grow in a appropriate conditions. Say, for example, some microorganisms require a lot of oxygen, lot of mixing is required. Okay, so that also again require a lot of input, the cost input into the process. Okay, so that we need to consider. And the microorganism should be resistant to predators like Delovibrio and bacteriophages. Bacteriophages, as you know, it's a virus that find its host in the bacteria. Okay, so in the virus, when the bacteriophages grow inside the virus, it lyses the cell like lysogenic and lytic cycle that you would have studied, and the microorganism will lose its property, or it will perish. Okay, so we should make use of the resistant microorganism for the production. So this strain selection is the first step in the upstream processing, or important step in the upstream processing. Once you did this very efficiently or correctly, appropriately, you're almost 30 to 40% of the job is done. Okay. Now we're slowly entering into the production process. So we classify the mechanical fermentation into upstream and downstream. Okay. So the fermentation process is being classified into two steps. Okay. Upstream, it has a three different classification, like producer selection of producer organism, selection of suitable fermentation medium, and third is the fermentation. So as I told, almost 30 to 40% job will be done when you finalize the strain. Okay, I can see among the three, first is the producer organism. Okay, and second is that once producer organism is fixed, and fermentation media is obviously very easy to design, and maybe you will be having the next two three, session afterwards. You will have the fermentation media optimization also. You'll be studying. Okay, so there you can get to realize how they're optimizing the media for a given strain. And finally, the fermentation, and we'll be discussing today what are different fermentation methodologies that are being adapted. And downstream processing is also part of your webinar, that you will be seeing how the product is being obtained from after the upstream processing, harvested, purified, and then marketed. Okay, so this is what I told. Fermentation industry is mainly driven. Otherwise, fermentation industry mainly driven means we are not maintaining these steps that I'm going to postulate now. Otherwise, fermentation industry won't be surviving for a long period of time. Okay, the cost and availability of feedstock, the feedstock should be year-round available, and the cost should be less, so that your production cost will be more. I mean, production cost will be less, and your profit will be more. Okay, and the efficiency of the microorganism. This again reflects the stability, genetically and physiological stability, and conditions required, and optimization about the microorganism, how it has to grow, what pH, what dissolved oxygen, whether mixing is required or not. And downstream processing, almost it also plays a huge role in finding the finalizing the product cost. Okay, downstream processing, because here very costly steps are being employed. Okay, depends upon the product also. So downstream processing also is an important aspect of the fermentation industry. So we'll be only considering the upstream process today. Later in the later webinars, we will be seeing about the downstream processing. So as I told, three different segments are being components of upstream process: producer microorganism, fermentation media, and fermentation. So producer microorganism, what you need to consider: selection of microorganism. So when you select it, you need sometime it may require the strain improvement also, either classical microbiology or genetic modification. You can do a strain improvement to make it as a more quality one, and then strain maintenance. So so that we can be able to maintain genetic and physiological stability, and then uh, we need to prepare a reliable inoculum time to time for its continuous production process. Okay. And second is the fermentation medium. So as I told, fermentation medium, depending upon the microorganism you selected, you can able to design a media that has the suitable or cost-effective carbon and nitrogen source. Carbon nitrogen source, say for example, all microorganisms prefer glucose, but when you give glucose as a carbon source, even though it is very easy, very convenient for the microorganism to utilize, but the cost of glucose is very higher considering the industrial perspective. So they don't give glucose as a direct carbon source. What they do, they choose some of the carbon source that are cheap in nature. Say for example, molasses. Molasses is a product or byproduct obtained from the sugarcane industries. The thick black colored molasses, still there was a 20 to 25% sugars are left. After our commercial sugar production, 20 to 25% sugars are still left as a glucose and sucrose. I mean sucrose. That molasses can be used in the fermentation industry as a carbon source for the other commercial product utilization. So now, when you have sugar industry also have an ethanol production industry. Why? The main sugar juice, they extract the sugar, and the byproduct molasses they divert into a ethanol plant where they produce the ethanol from the molasses, so that the waste is also being utilized, and they're having two products: one is sugar, other is ethanol from a single industry. Okay, this is what called sustainable fermentative industries. Okay, there won't be any waste product produced there. And finally, fermentation. Fermentation requires appropriate environmental condition like pH, temperature, aeration that are required by the micro. We need to optimize it because each microorganism has different physiological requirements. So say for example, some requires an acidic condition, some requires neutral condition, strict requirement of neutral condition, some require alkaline. It applies for the temperature also. We'll be seeing temperature with the classification like psychrophiles, mesophiles, thermophiles, different requires different temperature. Okay, and we need to, as I told before, when we apply fermentation process, we need to we need to target whatever products we are going to produce, whether it is a primary metabolite or secondary metabolite. Depending upon that only, we can able to design the final fermentation product for large scale production. Okay. Which the components of downstream processing, harvesting, and purification, and then finally the disposal of waste is also part of the downstream processing only. Okay. Let us slowly go into the how the industrial establishment looks like. It's just a flow chart that explains you that how a industrial microbiological establishment, that is a production establishment, have it. I can see the left side, the upstream process, downstream process. I have put a dark declassifier line there. Okay, so one is that culture related aspect. Center one is the raw material, and the right side is the physiological requirement. All three put together to have a proper fermentation process, and then the production happens. So these all put together in the upstream process. So to have some more diagrammatic representation. This is what happens in the real-time fermentation industry. Okay, as I told, once strain is selected, you need to preserve the strain for a long, considered period of time. So, if it is a real-time class, I could ask the question. Since it's live streaming, I could ask them because students' interactive is more important so that their knowledge is being enriched time to time. So, which technology being used for long-term preservation of microorganisms? Because we have a lot of storage methodology. Lyophilization is one of the technologies that are being used for long-time storage. Okay, we used to store the mother cultures. So mother cultures in the lyophilized condition so that it prevails for a long period of time. As I told, the Carlsberg industry been a prominent runner for more than 150 years. How they are maintaining the culture very safely without losing its stability. Lyophilization is one step technology where microorganism can be stored. So store not only storage, you need to revive it in the same stability. Okay, reviving of culture is also more important. How they revive? They revive in a shea culture, first in a petri plate, then the shea culture, then slowly the pre-seed fermenter and seed fermenter. You can slowly have a ratio. It's almost 10 times higher one compared to the other. Say for example, if the pre-seed fermenter is 1 liter, the seed fermenter will be the 10 liter. If pre-seed is the 10 liter and seed fermenter will be 100 liter. So it's almost the 1:10 ratio being put in the industries. Okay. So always the inoculum quantity is maintained as a 10 percentage of the volume. So if you are having a 100 liter reactor, sorry, 100 liter working volume reactor, so out of it, 10 liters should be your inoculum. Okay. So always prefix it, not more than, sometimes people go for 20% depending upon it, but 10% is the most recommendable percentage of inoculum required in the fermentation industry. That's why you need a feed fermenter separately apart from the production fermenter. You need a 10 liters of inoculum to feed into a production process that are working in a 100 liter. So always the fermentation industry require a seed fermenter. So for a 10 liter production, you need a 1 liter fermenter. That is called pre-seed fermenter. For 1 liter fermentation, you need a 100 ml shake flask. So you can now understand why I given this different configuration of fermenters because of the requirement in the fermentation industry. So production ferment is the ultimate where the media has been put up along with the inoculum for production of the metabolite of our interest. And the down portion you can see the plate and separation and extraction that we'll be seeing in the later part of the webinar. So today we are being restricted to the upstream process. Okay. Again, in upstream processing fermentation, even in fermentation can be classified into two different ways. Okay, so it's the solid state fermentation and submerged fermentation. It's not always the reactor when I shown here, right? The reactors that I shown, it's not always the liquid play a prominent role in the production process. Sometimes solid substrate is also being used in the fermentation industry. That's why we classify the solid fermentation into two different ways depending upon the substrate that we utilize. Okay, so that is solid state fermentation, submerged or liquid fermentation. Okay. So what difference it makes? Okay. So it depends upon the microorganism preference. It grows. Mostly the solid state fermentation. They go with the fungal. Okay, fungal microorganism. Okay. Why fungi? Fungi, you know the morphology of the fungi. Morphology of fungi is different as compared to the bacteria. What is the morphology different is that fungi grows by expanding its hyphae, hyphae filaments. Hyphae is the small filament like fibrous like structures that attaches to a substrate and it grows. Okay. Whereas bacteria is not like that. Bacteria does not much require the attachment to the substrate until and unless it is required. So most people go for the solid fermentation with respect to the fungi fermentation. Okay. And the submerged fermentation is not restricted to fungi alone, even bacteria also being used, but most predominantly or majorly the fungi fermentation are being done by the solid fermentation. Okay. And submerged, as I told, it's a normal conventional liquid fermentation using the bacteria, and even we can use the fungi also. But it depends upon the preference of microorganism that we use. Okay. This also you need to understand. Okay. So solid state fermentation. Let me give you a small glimpse about the solid state fermentation. Why solid state fermentation? Solid state fermentation, solid state that substratum like act in two ways. One is provides the support. Sometimes the substrate itself provides the carbon source. Say for example, when you provide the paddy straws as substrate for a fungi to grow, So the substrate, the fungi mycelia attaches to the substratum as a support. Also, it utilizes the components of the paddy straw as a carbon source. So it can act in the two way, okay, hand in hand. You can see the same image of the banana peel. Banana peel is one of the commercial byproduct obtained from the industries, banana processing industries. So they where to throw the banana skin, you can throw not throw as such, you can utilize in the fermentation industry where the fungi requires that. So you can see the fungi hyphae going over the banana peel as a mat over it for the growth. From that we can able to produce. So mostly the fungi are the important source for the enzymes. Okay, because fungi are all these saprophytic and other. Uh, it's a synergistic microorganism. It always depend upon a host. So when it depend upon host, it has to produce a lot of enzymes for its growth. Okay, so that's why fungi enzymes are being used in the commercial fermentation industry. Okay. So here also we need to consider it's not like directly giving the cattle feed. We cannot give the a solid substratum for the microorganism. Here you need to have some factors you need to consider for applying the solid substratum for the production of the enzymes for the fungi. Okay, so it starts from the moisture condition, particle size, period of fermentation, type and size of the inoculum, retreatment of substrate, and selection of suitable substrate and the microorganism. So these are the factors that are playing important role in production of enzyme by the solid state fermentation through fungi. Okay, so you need to consider these things into it. So as I told, sometimes substrate acts as only a support, and sometimes it acts as a nutritive supply also. Okay. So when I tell a classical example, you will understand. I will have 2 3 slides. I'm having one of the example, commercial example. Then you can easily correlate what is that solid fermentation. Okay. The support means sometimes only the polyurethane foam, vermiculite, inert fibers, only the microorganism attached to the support, and then it grows. Okay. Whereas the support and nutrient supply, your attachment, the support also provides the nutrient for the microorganism growth. Okay. So this is how the solid state fermentation industry looks like. They have either the plate and tray method, or it have a cylindrical fermenters are also big. Mostly ideal for filamentous fungi. As I told, only the filamentous fungi that utilizes the fungal hyphae for the growth. Even the yeast are also not prominently used for this type of fermentation process. Only filamentous fungi, Basitomycetes and Ascomycetes are being used. Okay. So, and it is cost effective. Why cost effective means it does not require much of electricity input, other physiological requirement like mixing, maintenance of temperature, or these are not required much in the solid state fermentation. That's why solid state fermentation is more cheaper, and cost of production is also more cheaper. Okay, so it's more cost effective in a way as compared to the submerged fermentation. Okay, primarily this solid state fermentation being used for the extracellular enzyme production, metabolites, antibiotics by the microorganism. I mean the fungi microorganism. Almost one third of the industrial enzyme produced in Japan is made up of SSF. Japan is more prominent in the fermentative technology. And other than that, now people are doing some cottage industry based ethanol production also being from the starchy substrate. Okay. Yes. So this one of the not here in India, it's in Japan. They do a fermentation of rice. You can see rice and wheat. You can see the white patches growing on the cooked rice and wheat. What is the white patch? Is the edible fungi. Okay. What happens? What is the taste of the rice? The raw rice. You don't realize the rice taste because it's so blunt. Okay. Because rice is made up of starch. Starch is a polymer, whereas starch contains glucose. So once the starch is broken down, you start to get a sweet taste because glucose is being released. So what they're doing in Japan is that to get a sweetened rice, what they do, they cook the rice partially, and they spread these microorganisms spores, that is the Aspergillus oryzae, usually called as koji, where they give a Japanese name called koji culture. They spread over it. Slowly it changes, the mold covers the cooked rice and the wheat. And what happens? These aspergillus grows on the rice and metabolizes the starch, broke down the starch into the glucose. So once glucose is released, slowly this rice start to have a sweet taste. So now they are employing for soya sauce production. Okay, you know what is soya sauce? that we use in soya fermentation. So the soya sauce is now through Koji only they are producing. Okay, this is being employed in the Japanese country, not in India, in Japanese country. And this is you can correlate. This is one of the prime example of the solid state fermentation. Now mushroom cultivation that we take in our daily to day life that are available in the markets that are nothing but the outcome of the solid state fermentation. So you can see the bag holder. You can see what is inside. It's a paddy straw been sprinkled with the media inside and along with the fungi spores. Okay. Once the microorganism grows, they make a small holes along the or around the bag to protrude uh, the fruiting body of the fungi. The mushroom is nothing but the fruiting body. The white patches are all the hyphae filaments that are covering the solid substrate. This mushroom is nothing but the fruiting body of the fungi. Okay, they harvest it and use it for the commercial purpose. It has been established that mushroom contain a lot of protein and lot of antioxidants. Even it is being used for the cancer cure too. Okay, in India we are using button mushroom, oyster mushroom. Most of you have been tasted in either in hotels or in your kitchen. Also, you would have seen. Almost 100% of this being obtained from the solid fermentation only. Let us go into the upstream processing again, especially in the submerged fermentation. Okay, so far we have seen the solid fermentation does not have much sophistication. Okay, there is no much sophistication. It's simple as that we do in villages, just like maintaining of proper temperature and requirement of infrastructure required in the industry like large trays, maintenance of proper moisture, temperature, and the substrate that we use. That are all the small requirements of the solid fermentation. That's why this mushroom cultivation being also used in the cottage industry. Okay, and there won't be much requirement of quality control here. Okay, so once you get a normal seed means the mold spores. That's also sufficient. That is sufficient for mass production. So now we are slowly getting into the submerged fermentation. So submerged fermentation, as I told, is a liquid fermentation. Okay. Mostly done in the reactor vessels. So how they are doing? They are doing in three different modes. One is the batch, second is the continuous, and third is the fed batch. Okay. So just I will tell a small, like analogy. Batch is nothing but a pressure cooker. Batch reactor is nothing but a pressure cooker. What you do, say for example, you want to cook a dal. What you do, dal means the dal grains that you want to prepare in the home day to day. What you do, you just soak the dal in water. You cut the pieces of onion and the tomato, put it into it, add some water, add some salt, and then put it in the pressure cooker and on the stove. And what happens after 4 whistles, you switch it off and take out the product. So that is the batch process. So you put all the requirements into a single reactor. After a considerable period of time or after the fermentation time, your product is ready. The same as the pressure cooker. You are not doing anything intermittently. Say for example, suppose you forgot to add salt during the second whistle. Can you able to open it and then add? No, you cannot able to do that. So because it's in the batch condition. Okay. So yes, batch does not require any or does not demand any intermittent addition during the fermentation period. Whereas continuous and fed batch is the alteration amount. Something has to be added intermittently or continuously. So continuous, the name itself indicates that a substance in the reactor is being continuously added and the substance from the reactor is continuously removed. Either of that way it constitutes the continuous process. And fed batch is the intermittent addition of the components into the reactor. Okay, not removal of product, just intermittent addition of component into the reactor. I will explain what it is. Very different. Okay. So as I told, just remember this analogy. I'm not going into much technological aspect of it as it takes much longer time. Okay. So batch culture is nothing but growing all the microbial growth cycle in a single reactor. Okay. The microorganism starts in the lag phase, grows, and then attains a stationary phase once the nutrients are being utilized, and then your product is ready. Say for example, these are being used in the mostly in the ethanol production. Okay. Ethanol production, even sometimes the microorganism itself product. Say for example, yeast baker's yeast production that we use in our day-to-day life, bakery yeast, that is also a commercial product. So what they do, need to mass multiply the yeast cells, right? So when the mass multiplier means we grow the yeast in a reactor. Okay, it does not require anything. And second and third, B and C are the components of the continuous culture. So we are classifying into a chemostat and turbidostat. So just split the word chemostat, turbidostat. Stat means what? Something stationary, and chemo means some chemical. A chemical signifies the growth component, I mean the carbon source, carbon or any rate limiting substrate. Okay, so when you add that component, the growth increases, or when you remove the component, growth decreases. That is called rate limiting chemical adding into the media. Okay, so in order to maintain, suppose a logarithmic phase. What is logarithmic phase? The microorganism grows in a logarithmic phase. What is stationary phase? When there is the depletion of nutrient, there won't be any growth happens. So you need to make sure that there won't be any depletion of nutrient happening to obtain the product of log phase. So you need to maintain the microorganism always in the log phase means you need to always feed the reactor with the carbon source, not considering its depletion. Okay, so that is called chemostat. Continuously adding the component that is carbon source without any depletion, so that the microorganism still prevails in the log phase, so that it gets lot of primary metabolites. Okay, turbidostat. Turbid signifies what? Turbid signifies the cell growth inside the media. Okay, here you have to maintain a proper population of microorganism inside the fermenter to obtain a particular product. Say for example, the stationary phase itself you can take. Stationary phase is where the micropopulation is higher, so that secondary metabolite is also produced. Okay, so we need to maintain the microorganism in a stationary phase. Okay, so depending upon the requirement, you are adjusting the flow rate or input and output from the reactor not to maintain this static condition, chemo or the turbidostat. What is fed batch? Fed batch is that adding the components of the nutrient in a limited quantity. Say, for example, when a microorganism requires 10 gram per liter of glucose, don't add 10 gram per liter of glucose as such. Just feed 1 gram per liter of glucose every 1 hour, so that the microorganism won't grow rapidly. Slowly grows. Sometimes this limitation of this. Sometimes when you add more of glucose, what happens? Feedback inhibition happens. More product is produced, the product itself come and inhibit the product. So I used to give a small analogy in the class. What means? So you are hungry, so you are so hungry, you didn't take food for morning and afternoon. Suddenly you have been provided with 50 laddus, okay, that are sweet in taste, made of ghee. So what happens when you are hungry, when you are provided with 50 laddus? So what you do? You start take the laddus one by one. So until you lose the appetite. Okay. So after losing the appetite, also you take one or two because of its taste. And after suppose after taking 15 laddus, what happens? You cannot able to take because you feel vomiting. So once you take the 16th or 17th laddu, what happens? Whatever you taken, you get vomited, right? That is called feedback inhibition. The food itself acting as the trigger to get it outside. That's a vomit. Okay, something like this is like a small analogy that you can always remember. Okay, don't think that I'm just telling this. So this always you can remember. Feedback inhibition play a prominent role in industry, especially in the ethanol production industry. When you provide more carbon source, microorganisms go very fast in the logarithmic phase. So what happens? More faster, more product is produced. The product comes in meet with the microorganism. Your microorganism will lose its viability. Okay. So instead of producing in a large quantities, give small quantity of nutrient. That is the fed batch process. So that there won't be any inhibitions happen during the growth. Okay. These are some of the advantages and disadvantages of collection and submerged fermentation. And in due course of time, you can read in the your syllabuses also. So I'll just tell the small kinetics. I'm not going because I always prefer kinetics to be taught in a board. It's very difficult for me to explain because as a biotechnologist, you should know the mathematical background behind this also. Okay, but today I'm just limiting myself towards the microbial growth with the model equations. With the equation one. Just 1 second, I have water. Yes, microbial growth kinetics. Why it is important? How it is going to govern your commercial production? So whenever microbe growth kinetics is being coming into your mind, you should have another correlative equation. It's a monad equation. Monad is a person who postulated this equation. Many of you would have seen mu is equal to mu m s by ks plus s. Okay, mu is nothing but the specific growth rate. Growth rate is the rate or the time at which the microorganism grows. Specific growth rate is the growth of a single microorganism. Okay, so the summation of all the microorganism is the overall growth rate of the microorganism in the given fermentation. Okay, so don't forget this equation. Mu is equal to mu m s by ks plus s. What is ks? Ks is the half saturation constant or the substrate concentration at which the microorganism grows at the half maximal level. So you should always make sure that your substrate concentration should not fall below this Ks value. Okay, Ks is nothing but the substrate concentration, saturation concentration. So why it is important in the commercial or fermentation industry is that your Ks value, that is a substrate concentration Ks value, should not fall below subcritical level so that the microorganism will be devoid of nutrients. Okay, that's why it's a design parameter for the fermentation industry. So the microorganism grows in a very logarithmic rate. That is the substrate growth rate. Like dx by dt is nothing but the growth rate of the microorganism and rate of change of microorganism, and mu is the specific growth rate, and x is the cell at any given time. Okay, as I told, mostly the bacteria undergoes the binary fission, so it grows in a logarithmic binary logarithmic phase. So we have a small terminologies being used in the growth kinetics. One is the generation. Generation is the time interval that is taken between the formation of one cell into the two cells. That is the generation. That is generation time. Generation time and doubling time. That's the two different. Closely observe. I'm telling about what's generation and generation time. Okay. So one generation, second generation means, suppose one generation means one cell will multiply into two cells. Third generation means the two cells will be converted into four cells. Okay, so that is the number of generation that does not have any unit, and time required for the generation is generation time, or otherwise the doubling time. That is the 20 minutes that we used to tell for equalize. That is the doubling time for the microorganism. So this is the general growth equation for the microorganism to determine how much generation it has undergone. We can able to predict it also. So n is the number of cells at any given time. N naught is the initial number of cells. And 2, what is the 2 signifies? 2 signifies the binary fission. Okay. And n is the number of generation. If you have a note, you can note it down. So last 3-4 years, I've been into SRM for the past 3 years. We used to conduct special gate classes for the students. Okay, if you are an engineer to pursue a higher education in the top elite institute, you need to qualify the gate. Okay, so gate, you have two different gate: gate in life science, gate in biotechnology. Okay, gate in life science is morely of a theoretical aspect, whereas the gate in biotechnology some involves the engineering aspect. Okay, those who are listening, who are in the engineering background, even this is not, it is also being prominent in the science background also, because it's not much of rocket science. Okay, so this is just a growth kinetics. So this type of equation is helpful for tracking the national level examination. That's why I asked you to kindly note these things. Okay. So you can find it in any textbooks. I'm having the reference book also. I'll show in the end of the slide to refer whatever reference book you can refer. So this is the growth number of cells predicted at any given generation. Okay, so we have a growth phase, stationary phase, and what happens once the stationary phase attains, there is start to happen that there is a start of happening of cell death. How the cell is growing, the opposite of it also happens. Okay, so that is the cell lysis because of the non-availability of nutrient, or because of the antibiotic produced by the other counterpart. So cell death also happens. So what happens at time? So mu is the growth rate, means KD is the death rate. Okay, kindly correlate with each other. When mu is the growth rate, KD, that is the death constant. Okay, KD is the death rate. Okay, when that is being signified as a negative symbol. Okay, minus signifies the death is happening. Okay, so how the cells are being n is equal to nr, like 2 to the power of n growth is being signified. You can see here how the death is being calculated. N is equal to N naught e to the power of minus kd into T. Okay, this is the death kinetic equation. So here, where it plays a significant role is that during the sterilization process. What is sterilization? Sterilization nothing but killing of microorganism in the nutrient media. So before you start a fermentation process, you need to sterilize the media, right? So why sterilize? Because the media contains it's a raw like liquid or components that may contain unwanted foreign microorganism. When you put the pure culture, it may get contaminated and your product also get contaminated. That's why before you start a fermentation process, you should sterilize it. These this death kinetics plays a prominent role in calculating the what type of sterilization time or sterilization condition required, what time is required for sterilization to reduce the time. So here you need to kill the microorganism, I mean unwanted microorganism. Don't take it the water microorganism, it's unwanted microorganism. Okay, so this is the death rate. So this is some of the growth associated products. So as I told, some of the products are growth associated, some of the products are non-growth associated that are mainly produced in the secondary phases, right? Secondary metabolism, as you can see. Some products are produced in both in the growth and the non-growth condition. There is a mixed growth associated product. Okay, so you have a rate equation between each governing as depending upon the product produced. Okay, so you have a yield coefficient y-axis, that is amount of microorganism grown with a given substrate, YPS amount of product produced with a given substrate. These are some of the significant growth parameters, kinetic parameters in the fermentation industry. Okay, you need to have a thorough read about these kinetics. When you understand the basics, you can easily understand the kinetics also. And another one important aspect is that this temperature. So temperature is directly proportional to the growth. By increasing the temperature, you can able to achieve a higher growth, but not very directly proportional. There is a limit also. Okay, so that's why they classify the microorganism depending upon the temperature as psychrophiles, mesophiles, and thermophiles. Say for example, 20 to 50 degrees Celsius is the mesophiles. So when you increase the temperature from 20 to 50, the microorganism tend to grow faster. Okay, within this range only. Once you it reaches 50, once you raise the temperature 50 to 60, if it is mesophilic, it start to die or start to reduce the production because it comes under the thermophilic condition. Okay, so that is being governed by equation called Arrhenius equation. Many of you would have known in the school also, and here also it is there, Arrhenius equation, where what type of temperature you need to employ from the range. Okay, because we have only the range, or we know only the mesophile. We don't know the exact temperature where the maximum product is obtained. So using this Arrhenius equation, we can able to deploy a proper temperature for the maximum production of product from a given microorganism. So you can see here on a classical example of the net growth rate. Net growth rate dx by dt is equal to mu minus kd. Mu signifies the growth, and minus kd signifies the death. So net means what? Net growth means what? The, both the growth should be higher, the death should be lesser, so that you have a constant amount of microbe population inside. Okay. So in order to have a very efficient growth, we cannot avoid death inside any inside the rate fermenter. We cannot avoid. That's why we need to consider this minus kd. Okay. So, but it should be minimized. KD should be minimized. Okay. So it depends upon the type of product that you are producing. So this is the net growth rate of the microorganism in the fermentation process. So environmental factors, the dissolved oxygen, pH, the salt concentration, that is salinity, are all play a significant role in the microorganism's growth. Okay, the salt concentration. Sometimes some microorganisms require extra salt, micro and macronutrients. Okay, dissolved oxygen is required very highly for the some of the microorganisms that are producing the organic solvents like acetic acid, citric acid, and those things. Okay, whereas ethanol is an anaerobic process that not mainly require the dissolved oxygen. Okay, D word signifies the dissolved oxygen. Okay, these are the two prominent books that are available for bioprocess engineering. It's very basic book. You can note down the many of you know, but if you are in the early stage of biotechnology, like first year, second year, like that, you can note down this. These books are helpful for your basic level understanding of microbial growth kinetics, and even you can able to crack the examination by having a thorough understanding of concepts of these books, Doran, Tudor, and Tarki. It's like a Bible for the bioprocess engineering. Okay. Yes. Now we'll slowly go. So far, you would have understood like what happens in the fermentation industry. Start from the strain selection, strain maintenance, fermented medium, and then the fermentation. But since you are going to study about the webinar in the next about the fermentation medium optimization, I didn't cover it. You'll be studying, learning in the next webinar. And these are the important aspects of it. So, what main thing is that you need to reduce the cost of production so that you can able to run the industry in a profitable mode. Okay. Now there are recent advancements happening around not only in fermentation industry, in general manufacturing industry, the innovations are happening. So many of you would have not aware of this industry 4.0 because it's so new to biotechnology. Whereas the automobile industry and other electrical and other industries know this concept of industry 4.0. Whereas for us it is very new. Uh, Even if you search in the net also, you might not get. This is my own understanding and my own deliverance of combining of biotechnology with the recent advancement in other industrial aspects. Why can't we have a hybrid production technology? Okay, so Industry 4.0. What is Industry 4.0? And before that, I just slowly have a uh, why where from it started. So lot of advancements happen in recent years. In the almost last uh, 10 years, many advancements happened. Like when I did the BTech, that time different scenario I was there. Now when I'm now in the present situation 2020, the different things are there. Okay, you're starting from a social platform to the usage of gadgets, many things are changed. Okay. Similarly, the industrial production technology is also being emergently changing, but not in a rapid manner. Okay, because when everything is changing, that also has to be changed, right? So what are the changes we expect in the industry? What at what it has been attained? So there is a recent advancement called cyber physical system. Okay, so cyber physical system, to put it in a simple layman term, it's like a robotics. So what robotics does? So you does not require any manpower there, or it does not provide it does not require any real person there to exist. Okay, so yeah. So what happens? The robot's does everything. It analyzes and it gives you output, right? So robots, if you give a command, what happens? It does its job and give you the output, right? So it's something like a cyber. Cyber is nothing but utilization of the sensors and other computerized aspect to handle some physical activity to get a output. Okay, it's been done by the three C's. One is computation, control, and communication. Computation is nothing but the programming, the coding, and those things. And through that, you can able to communicate and control a system to get a output. Okay, so this is playing a background behind this industry 4.0. Why this industry? What is this industry 4.0? Is that incorporation of this cyber physical system into an industry to get a more more sensitive or more reliable product with a short period of time without much of error. Okay, because what happening is that when we are doing in a manual mode, there is a lot of happening. There is a lot of chance of error. Human errors are there, manual errors are there, equipment errors are there. Okay, what happens? It ultimately reflects in the cost of production. So they are incorporating. Say for example, the car manufacturing industry nowadays, they slowly started to reduce the manual mode of fabrication. They slowly they have incorporated the robotics so that the error cost during the production has minimized. What happens? Sometimes you would have read in the newspaper. Ford has recalled the 2,50,000 cars that are having the malfunction in the steering gear during the manufacture process. So what happens if they need to repay the amount to the consumer, or they need to give the new car instead of that? So it's actually a damage to the industry economics, right? So this happens in the biotechnological pharmaceutical aspect also. So you can see a lot of I should not name the industries. It's the common forum. So there are a lot of uh, mishandling errors happen in the industries, the biological sector also. Okay, so they have recalled the products from the market and they provided the compensation too. Okay, so in order to avoid such errors, not to avoid such economic backlogs, we need to implement some high sensitive cyber physical system. That is nothing but the industry 4.0, fourth generation. So far we have seen about, now we are in the 4G. Okay, this industry 4.0 was been prominent in 5 years back in Germany. Okay. So it's mainly done to have a interoperability. Interoperability means interpolation. So how much of product that can be produced in a given substrate. Okay, interpolation or extrapolation. Okay, so we can make it out in a we can assume assumption what amount of product we can able to get in the future predictions. Okay, virtualization, what happens in a virtually decentralization. Decentralization is that not happening in a single space, and each component of the fermentation industry can happen in a different location, being handled by the Internet of Things. Internet of Things, IoT, these are the buzzwords nowadays. We biotechnologists does not aware of this, and we should do this, implement this recent technologies into into there. Okay, slowly agriculture started to adapt this one. Agriculture, since it's a biological aspect, but the pharmaceutical industry and other biological industry has to adapt such recent buzzword, Internet of Things, controlling the things in a through cloud computing. Okay, and real-time capability, service orientation, and modularity. This is smart factories. Smart factories means what? Smartphone. Your smartphone that you are having today is is just a culmination of 60 to 70 things that are found that are being merged into a single smartphone. Say for example, alarm clock is not alarm clock. You don't require a watch. You don't require a calendar. You don't require a map. Okay, you don't say there are many things that are being converged into a single mobile device gadget, right? That is smartphone. That's similarly different aspects of recent advancement being imbibed into a factory to convert into smart factories so that your error is being minimized. Right? So, what are the building blocks of Industry 4.0? So, I'm just telling this aspect. Just you yourself correlate now, or else in the future, you just correlate how we can implement these things in our biological industries also. Autonomous robots, okay. So, robots that able to do the things precisely. Simulation that I told, predict simulation means if the temperature is 60 degrees Celsius, what will be my product quality? If the temperature is 40 degrees Celsius, what might be the product quality? Okay, the simulation and prediction. Okay, so you not necessarily do the experimental in the lab, get the results, and then doing it takes a lot of time. So by reducing the time, we can reduce the cost also. Simulation helps in a great way. Okay, horizontal and vertical system integration, it's not much into it, into our thing. And industrial internet of things. Industrial internet of things is that communicating different industries with through the cloud computing. Say for example, I have a plant one in Chennai, I have a plant two in Mumbai, so I need to have a simultaneous production in the plant one and two also with the same substrate. Okay, so these two factories can able to communicate with each other through the cloud computing, that is called Internet of Things, to able to produce the same product quality in Chennai and Mumbai also. Okay, so that is that industrial Internet of Things. So different location being integrated, and so that you can able to handle the things in a smooth and very less time. Okay. Cyber security is not in our thing. Yes, sometimes it is different by because sometimes people can able to hack the formula. Say for example, Pepsi and Coke. Why they are more prominent? Because they have their own Pepsi formula, Coke formula. Even though it tastes similar, but it has its own formulation, right? So sometimes what happens if we go to the industry 4.0, if they hack the formula, somebody can able to manufacture. So cyber security is more important if you want to implement the industry 4.0. That is the hacking thing. Additive manufacturing. Additive manufacturing means doing manufacturing in Chennai, doing manufacturing in Mumbai, so that you have overall increased product quantity. Now, I correlate now the present scenario. They are telling that in August 15th they are going to release the vaccine for COVID, right? Again, vaccine means it has to be given for a whole population of India. The whole population of India has to be administered. Means that much production should also happen. So a single industry cannot able to produce that much vaccine. So what happens? A diversified industry, different locations of India, if they have the plan, they have to have additive manufacturing from one place so that the quantity of demand can be met. Okay, second is the augmented reality is not much into our own aspect. big data analytics. The big data analytics is mainly prominent role in a biological aspect in terms of the sequencing, like sequencing of microorganism, human genomic sequencing. Like big data means lot of data is required. Genomic data are very big in nature. You need a supercomputer to analyze it, right? So even it can play a prominent role. So these are the components that are playing a significant role in the other industries. Why can't we implement in the fermentation industry also to improve the product quality and the quantity? Okay. Now we are facing now the world, each and every one of us is being looking for a vaccine for a pandemic, right? So only the biotechnology industry that are having a uniform quantity throughout the world can able to meet the demand to overcome this pandemic period. That's why I thought to incorporate this aspect into today's lecture. So, what are the potential implications of this Industry 4.0 in fermentation industry? It can be robot-assisted production to reduce the manual human errors. Predictive maintenance. Predictive maintenance is that yes. I'll just tell a small correlative. Recently, there was a huge emergence in the smart vehicles. Smart vehicles means the car being incorporated with sensors. Suppose your four tires are there. In one tire there is a less air, means the car will alert. Now we have only the fuel alert only. But the smart cars are being incorporated so that it maintenance also the car itself take this car to the maintenance. Car itself tell its healthiness to take for its maintenance. Similarly, in industry, nowadays only an engineer has to be there to look into the maintenance of each and every equipment's precision and performance. Whereas these sensors can able to predict this maintenance requirement so that errors can be overcome by having the predictive phenomena. Okay. Additive manufacturing of complex parts. This is not complex parts means this is for automobiles. For our industry, like additive manufacturing means compound one is produced in Mumbai, compound 2 is produced in Chennai, and these two can be combined in Hyderabad. Okay, this is additive manufacturing. Okay, uh, the big data driven quality control, that is, as I told, mostly our genomic work is being employed. A smart supply network. Smart supply network means your product starting from the production to the market end user, you can able to track it. So you can have a barcode in your packet, and they can track you how much of packets being sold from the shop, how much of packets has been went to your home. You can able to track it also. Okay, nowadays the technology has come up to this level. Okay, so what impact it will have? It will boost the economy, overall Indian economy or national economy, boost the business directly or indirectly, and both national and global. As recently the Prime Minister has told that India is going to be the world prominent role and leader in this pharmaceutical sector. Mostly, I think it might be this type of incorporation of advanced technology, and ultimately, what happens? It's going to serve for either an individual and society together. To have a correlation of today's scenario is going to serve both the individual and the society purpose. So it has I started with the fermentation industry, I ended with the society, right? So you can see the interlink with each other. This is called the chaos theory. It has each vibration has its own aspect at the other end of the earth. Okay, so you need to apply it. So growth, productivity, employment, labor substitution, the nature of work. So yes, you can ask a question. Yes, if you have replaced the robots for manpower, where will get the job? Yeah, right. Yeah, you need to rethink in a way. So you need to be a um, person who can able to handle those robots, because robots cannot able to run on its own. So you should be the control of that robot, right? So you need to upgrade your knowledge in that scenario. Okay, it's not like manufacturing skill only. You need to upgrade your skill depending upon your recent advancements. Okay, so just break down the wall around you as a biotechnologist. Yes, lab works are more important. Just try to learn all the aspects of current technologies, how you can incorporate into biotechnology so that it can be a advanced version too. Okay, so this will impact the businesses, that is customer expectation, the products, collaborative innovation, and new operating models. Is about the business improvements. And yes, as I told, it should or it must have a national and global impact. Nowadays, FDI means foreign direct investment. It's not necessarily, yes, Make in India is there. Even a US company can come and start a company in India with the technology available. So if you are skilled enough to able to manage such advancements in our field with biotechnology knowledge, you will be the uh, you will be selected in the interviews, and you'll be prominent leader in this area. Okay. And society, there's a lot of society. It's like sometime previously mechanical engineering means only boys only will select, right? In those days, mechanical engineering means you can see only boys in the class, but not. Why not a girl can not able to study the mechanical engineering? Nowadays everything converted to sensors, right? So even a girl can also study a mechanical engineering about the sensors, and she can be a role in the mechanical engineering aspect. Yes, these advancements in sensors, industry 4.0 can able to overcome this inequality happening in the industries. Okay, every industry can also employ a women also in the place of man equally, or the whatever the proportion is been expected. Okay, and individual connections, identity, morality, ethics, all being built up with the time. Yes. So this is, as I told, previously 10 years back, the interviewer expectations are different from you when you go for an interview. Whereas in 2020, when you go for an interview, your expectations are interviewer expectations are different. Okay, if you are able to see the slides, you just note it down. What are the expectations of nowadays? What type of skills are being expected from you? Don't always put a blame on the courses or the field that you study. Sometimes what happens? Do I have a scope? Do I get a job? What type of job? How much of salary I get? How much of package I get? They compare the other degree with our degree. They compare other job with our job. Okay, it's not like that. It's a requirement demand from each other field. Okay, so you should make sure that you able to meet these type of advanced technical skills in order to meet the current demand in the industry. Okay, so these are the skills that are being expected in 2020. It's been the report of the future job report. I also World Economic Forum. I took these from a source only. Okay, so just you should make sure that relevance of your skill towards the current technology, right? Yes. With this, I complete my session. I hope I have given a brief about upstream process, analytics, and the innovation. Thank you. If you have any queries, you can.
Thank you, Samuel Jacob, sir. Now I kindly request Dr. Mahendra Kumar, Assistant Professor, Biotechnology Department, to take over the discussion session.
Yes, sir. Yes, ma'am. Thank you very much, sir, for your wonderful and very elaborate and detailed presentation. Uh, I shall pop up the questions asked by the participants. Dear participants, if you have any questions related to sir's presentation, you can type in the chat box.
Okay, I can see here. Are there any proteins in fermented products which are helpful for the curing of internal wounds? If there, can we produce them in large quantity? It's asked by Chatla Abhishek.
Yes, yes, sir.
Yes, Abhishek, you have put a question. Um, there are some proteins that are being used in the pharmaceutical industry. Say, for example, interferons. Okay, interferons are all the proteins that are naturally produced by the cells. Say, for example, it's alert cells. Suppose one cell is getting affected, means the affected cells produce some protein called interferons, so that it alerts the next cell to overcome the infection. Okay, so they identified this thing in the laboratory, and they thought that why can't we produce the interferon in the large quantity so that it can be used as a vaccine too. Okay, so yes, interferons are all being produced. Interferons are also a protein. It's used for curing the infections. It's produced through recombinant technology, the large quantity. Danisha Mohan. These are used as the starter culture for fermented food. So, what is the role in commercialization of fermented food? Yeah, fermented foods. What is their role in commercialization of fermented food? Yeast, as you know, yeast we use two type of yeast, right? One is for alcohol production, and another is the baker's yeast that we use for bakery industries, right? So you know the commercial aspect of both the ends. Okay, um, the bread production industries, the breeding industries are also surviving it. Yeast are more important. As I told, a single Carlsberg yeast number one can able to survive an industry for 150 years. Okay, why can't we why can't we create a yeast that could be able to carry over that bakery industry? So I'll tell you a classical example. In 2010, Government of India, Ministry of Food Processing Industries, uh, they have started an initiative. Each country has their own uh, food, right? Say for example, if you take Italy, pizza is their national food, okay, or their geographical indication food. If you take Chinese, noodles, soups are there, okay, and hot dogs from USA. For India, if you take, they'll tell the different things. Somebody tell chapati, somebody tell dal, somebody tell rice, somebody tell idli, dosa, and it's been done. Yes, yeah, it's been discussed in Parliament in 2009. In Parliament was discussed, and Minister of Food was recommended that idli, idli from the idli batter as the geographical indication of Indian food. Okay, because it's been researched and it's been told as the idli is being nominated or voted as the best breakfast in the world. In the world, you can go on certain there. Okay, but again, idli has a different taste and consistency from region to region. If I take idli in India, sorry, in Tamil Nadu, it is different. If I take idli in North India, it is different. Why? It is depending upon the proportions of the rice, lentils, and the yeast culture that we use. It's actually yeast only. Okay, so why can't we have a scientific aspect, the background behind the idli production? I was doing my MTech project in Indian Institute of Food Process Institute in Tanjavur, Food and Crop Processing Institute. There, that project was there. I was there that time. They conducted a competition with housewives only. The housewives who prepare a good idli will be given prizes. Why it is that, as you can see, in curd they add a small quantity of curd to the milk to convert into curd. Okay, whereas in idli, in batter it's not like that. After the crushing of the batter, they used to mix it with hand only. Whatever the microorganism in the hand, it get fermenting. So why can't we, as you told, if you could able to create a common culture for idli batter also, it can be a uniform idli quality throughout the country. Okay. So why I'm telling is that you told about yeast. Okay. The idli batter also being fermented by yeast. So you can do a research also. So you can have a research orientation too. Why can't you prepare a separate yeast strain and patent it if you could and do a commercialized one? Like we have baker's yeast, but we don't have any idli yeast now. Okay, so I don't know the last person's name. You can patent it too. And third question, Tarun, viruses used in fermentation technology. Then what is the role of viruses in fermentation? Viruses are not used in the fermentation technology. No, I didn't told the viruses are being used. Viruses can be used as inducers for some, uh, as I told, interference. Okay, interference are being induced by the viral infection only. Viruses can be used as antigens for producing antibodies. Okay, viruses not being mass produced, whereas it can be used as antigens for mass production of antibodies that can be commercialized. How can I calculate ammonia hydroxide consumption for pH maintenance of the E. coli fermentation process? Ammonium hydroxide consumption means NH4. Uh, you have a biochemical methodology, right? So ammonium hydroxide, ammonia you can able to estimate by same methods. So you have an initial and final, right? From that you can able to calculate the consumption percentage. The maintenance of pH, it depends upon how much you're adding, like whether it don't affect the E. coli, how much you're going to add. So it's optimization only. Maybe the next 2 3 session, you will be having the media optimization. I don't know. Ma'am may tell relatively. So there you might be enriched with the knowledge of how you can able to calculate the ammonia hydroxide to be added for pH maintain. Yes, Abhinandan Prakash Patel. Is fed batch culture better than batch culture? Yes, obviously. Yes, it has been proven that fed batch culture is much better than batch culture in many ways. I cannot generalize it, but again, it depends upon the product, depends upon the microbe. As I told, microorganism behaves well in the fed batch culture because of the feedback inhibition or the substrate. The substrate inhibition kinetics also play a role. Okay, so why can't we have fed batch? Because the product is going to be same. Why can't we have fed batch system instead of batch? So batch is actually a, a preliminary thing before the advent of the mortars, fermentation technology. The batch is being done. Now people are doing fed batch. Yeah, fed batch is more dominant than batch. Nisha Mohan, what is the effect of uh, fermentation for byproducts on growing bacteria? What is the effect of fermentation byproducts of growing bacteria? That's what I told. The fermentation byproducts, uh, it should not be an inhibitory one. Okay, so if it is inhibitory one, you should be constantly remove it from the fermentative media. That is by through continuous mode of removal. So whenever there is a Archimedes principle, you know, right? So the volume should remain constant. When you add something, you need to remove something from the media, right? So you always keep on dilution. One thing I forgot to tell is that dilution rate that play a significant role in the maintaining chemostat and thermostat. So you need to dilute the toxic products, byproducts, so that microorganisms won't get hampered. Sir, can maintain the quality of fermented foods. Uh, quality maintain the quality of fermented food. I think the sentence was, yeah, maybe I can understand how we can able to increase the shelf life of the fermented foods. Yeah, shelf life is an important aspect. Shelf life can be improved by addition of preservatives. That preservative also being produced by the fermentation industries also. Say for example, inulin. Inulin is one of the components that are produced by the fermentation process. It's a modified, I think, amino acid that is used as a preservative. Okay, so it can able to prevent the spoilage for a certain period of time. There are many preservatives also are there. Even citric acid also a preservative. So that can able to maintain the preservative. The pickle is what? It is being not undergoing spoilage. Why? Because it has a acetic acid inside, right? So the vinegar, this will be some preservative. Yeah, through fermentation can able to preserve the quality, the spoilage. What causes rice water to get ferment, and what is the exact use of it? Rice water is what you like you as you know rice contains starch. So what happens? It's a starch water. Okay, so starch is a carbon source. You have a microbes in the air also, right? You know we have the microbes in the air, so both spoilage and beneficial microbes. Mostly the spoilage microorganisms are there. Okay, so what happens if we keep the amylolytic microorganisms might have been spoiled the rice water and they can eat. What I can tell you that in Japan, they are using. I showed the koji rice, right? They are using the wine fermentation. They use a white wine. Usually, wine is being consumed the uh, purple color, right? Because of the color of the grapes, right? Whereas grapes are not much available in Japan, but they need wine. So what they do, they ferment the rice water, as you have mentioned, okay, to produce the white wine. They add a uh, they add again taking white wine that does not correlate to your mind because white always looks red color. But if it is white color, your mind perception won't allow it to take because wine should be red, right? So what they do, they add a colorant that is produced by a particular fungi called monoscus that produce light pink. You can go and search it in it. So they add that fermented rice to molasses. So the rice water itself gets converted into red color, and the water contains the ethanol after fermentation. So it's being used in the wine fermentation from the rice because always the grapes are not being used. Other starch sources also being used for wine fermentation.
Thank you very much, sir. I think that's all. All the questions have been answered. Over to you, ma'am.
Thank you, Mahinder sir. Now I'm just rendering my a very short vote of thanks to all of them. I take an immense pleasure in rendering my heartfelt thanks to all the dignitaries, Mr. Manjit Singh Nair, General Secretary and Correspondent, in his absentia, to support in all our endeavours. Dr. N.J. Raghunathan, Principal of Guru Nanak College, for his continuous guidance. Both our Vice Principal, Ma'am, Deans, and all the participants for attending the sessions. My special thanks to Dr. Samuel Jacob for your interesting and informative session. I hope everyone will agree with me that speaker has very well highlighted that waste of one industry is a raw material for the another. Thus, fermentation technology truly supports for sustainable development and in mitigating the industrial waste, and also supporting for the circular economy. Thank you for sharing the importance of the development of softwares for the better fermentation processes and the production. As Sir has mentioned, there are good more opportunities for the students and for the researchers in the future. So let's all get innovative. I hope the session was useful to everyone. I can see encouraging and positive comments from all of you. I once again invite you all for the forthcoming sessions in this webinar series of fermentation technology. Thank you all. Now over to Dr. Mahendra Kumar for a few more instructions.
Yes, ma'am. Thank you very much, ma'am. Dear participants, please submit your feedback forms, which have been posted in the chat box. And I also welcome you to the third session, that is tomorrow at 11:30 a.m. Ma'am, is my screen visible? Ma'am, is my screen visible, ma'am?
Yes, sir. Yes,
Yes,
sir.
yes, yes. Visible.
Yes. So tomorrow's presentation, third session 3, will be handled by Dr. Veena Gayatri, who is serving as the Assistant Professor, Department of Biotechnology, Stella Maris College. She'll be talking and enlightening us with the topic media formulation for lab to industrial scale. The session will start at 11:30. The YouTube URL link for the same will be shared to you via your registered email ID and through the WhatsApp groups created exclusively for this purpose. I request your cooperation and participation in all the forthcoming sessions of the webinar series on fermentation technology. Thank you very much.
Thank you, ma'am. Thank you, sir.
I request all the participants to look into the chat box for the feedback links. The session 1 and session 2 feedback links have been posted.
Can we end the session, ma'am?
Yes, sir.
Okay. Sir, thank you so much, sir. Samuel, sir.
Thank you. Thank you, sir. Thank you, ma'am. Thank you for inviting. I hope the session was good. I didn't see the feedback. You will send the feedback to me?